Okay, so we'll start chapter 9, which is going to be conducting experiments. And okay, so in regard to experiments and selecting research participants, we can draw participants through probability or non-probability sampling. We've talked about those before. Probability sampling is going to be based on randomization. Non-probability sampling, this would be like us using a convenience sample and whoever is available. But when it's important to accurately describe the population, we need to use probability sampling or that randomization. In research that's more interested in testing hypotheses about behavior, we can find participants more easily using non-probability sampling methods like a convenience sample. But in general, by increasing sample size, it's also going to increase the likelihood of statistically significant results as well. That's going to increase the power of the research that you're conducting, whether that is going to include a probability or a non-probability sample. So that's why it's integral in order to have a large sample size or as large as possible. So first, in regard to manipulating the independent variable, we have to set the stage. We have to provide participants with the information necessary for them to provide their informed consent. And again, we talked about that including the benefits and risks, any of the ethical considerations, the procedures, any sort of compensation, any debriefing, and then what the study is basically about. And then we need to create a setting in which it's going to be amicable for participants to participate in. Now, how the variable, again, that's going to be the independent variable, is going to be manipulated is going to depend on what type of variable we're talking about, the cost, whether it's feasible, and also the ethics of the procedure. So these are the types of manipulations. We have straightforward manipulations. This is when we use instructions and other stimuli to manipulate an independent variable. Whereas staged manipulations, we're going to stage events during the experiment to manipulate the independent variable successfully. Now, these are going to be done for one or two reasons. We try to create a psychological state in participants, so we have people in a certain frame of mind, or to simulate a real world situation that may occur in the real world. And staged manipulations are going to employ usually a confederate, sometimes called an accomplice. We discussed this when you can think about Milgram's experiment, you can think about Ash's line experiment as well. In these studies, again, there's going to be somebody that is participating that is actually not a participant, but they are in on the study. And again, you can look at Ash's conformity experiment. So again, here's Ash's conformity experiment like we talked about. Now, the strength of the manipulation, a general principle to follow is to make the manipulation as strong as possible in order to get the fullest effect. And this is the potential amount of impact of the independent variable on the dependent variable. It's important in the early stages of research, and there's a couple of considerations. The strongest possible manipulation could entail a situation that rarely, if ever, occurs in the real world. So it may include something that is much more extreme or powerful or emotionally viable than what you might actually feel or experience. And the manipulation should be as strong as possible, again, within the bounds of ethics. Okay, so again, the dependent variable, how much somebody likes something, the independent variable, how similar attitudes are. And you can see here there's a positive linear relationship. And as attitude similarity increases, so does the amount of liking. Now, when it comes to the cost of manipulation, one that uses a straightforward presentation of written or verbal material is going to be less costly than like an staged experimental manipulation. And so, again, with that straightforward manipulation, you might just be presenting participants with a narrative that they need to read, and that's going to prime them in order to behave or have a certain emotion in a specific way. That's going to impact that dependent variable. Okay, now the ways in which researchers may measure the dependent variable. Okay, so again, participants are going to be exposed to an independent variable that has either been um, manipulated or not manipulated, and then the dependent variable is going to be measured. One way in which this is done is a self-report. This is Participants are going to describe what they're experiencing. The most commonly used rating scales are those with descriptive anchors or endpoints. This could be anything. Again, we talked about strongly disagree, disagree, neutral, agree, strongly agree. So again, we're using basically like a survey. Behavioral measures, this is going to be the direct observation of behaviors. And that observed behaviors, again, they have to be quantified in terms of like how often maybe somebody is smiling, how often somebody is laughing, how quickly they react to some sort of stimuli, and how long they are behaving in a specific way. This is going to be observed by the researcher. And again, sometimes the behavioral measure, it's not an actual behavior, but a behavioral intention or choice as well, maybe to make a certain action. Now, what you commonly will see, again, we see these types of experiments done, but you'll also see ones with physiological measures that are going to be a little bit more complex. And these are going to be recordings of responses of the body. So you might have the galvanic skin response. This is going to be use the electrical conductance of the skin, and EMG, electrical activities of the muscles, EEG, activity in the brain, MRI, scanning body structures, including the brain, to create images, and an fMRI, measuring blood flow to areas of the brain to create an image of electrical activity. So these are also techniques that can be used to measure the dependent variable after participants are exposed to the independent variable. Variable, again, that dependent variable can be measured in a variety of concrete ways. If it has the same effect on several measures of the same dependent variable, confidence in the results is going to increase. And it's useful to know whether the same independent variable affects some measures but not others. And so you might, in an experiment in which, you might be doing an fMRI scan in order to determine what effect something has on a certain area of the brain. Instead of that, you actually might need to um, use an EMG. Maybe it has something more to do with muscle contractions, but this is where you have to determine, okay, what is the best way to measure the effect that we should be seeing? And research may also be interested in the effect of an independent variable on several different behaviors. Maybe you do have an independent variable that's going to affect muscle contraction, as well as electrical activity in the brain. So you might be using different measures for that dependent variable. All right, so additionally, okay, in measuring the dependent variable, the sensitivity of the dependent variable is going to make a difference as well. It needs to be sensitive enough to detect differences between groups. This is going to be important when we measure like human performance. So there's a ceiling and floor effect. The ceiling effect is going to be an issue where the independent variable, it appears to have no effect on the dependent measure because participants quickly reach the maximum performance level. And so they reach it so fast that you aren't necessarily able to get a reading or a measure fast enough. There's also the floor effect, and this occurs when the task is so difficult that hardly anybody can perform well. So as a result of that, you're not going to see individual differences because the task at hand was too difficult. Also, when it comes to measuring the dependent variable, the cost is going to make a difference as to what you can afford. Pencil and paper self-report are pretty inexpensive, but then if you need to do like fMRIs, that's going to be incredibly expensive. Also, controlling for participant expectations, demand characteristics, and these are going to be the features of the experiment that could inform participants of the purpose of the study. And so if participants are going to form expectations, this is going to result in possibly response bias. And this is when participants are going to base those expectations on what they're actually experiencing. And so sometimes deception may mitigate this effect. Again, you need to avoid that so you don't have that response bias. And so there might be filler items. These might be items on a questionnaire that have nothing to do with what you're actually studying. And this is in there in order to disguise what the dependent variable is that is actually being measured. Also, by administering an apparent treatment, it might be sufficient and a sufficient cause to an observed improvement in behavior. This phenomenon is known as the placebo effect. Again, so you have somebody in a control group and you appear to give them some like an antidepressant pill, but it's really just a sugar pill that can result in a placebo effect. And so to control this, researchers use a placebo group. And so this is um, a group of participants in a study who receive an inert substance instead of the experimental drug. So if the placebo group improves as much as the experimental group, all improvement could be caused, by the placebo effect. And these placebo control groups, they do have ethical implications. Those receiving the placebo are due the actual treatment if proved effective. So the ethical issue here is that we have to make sure that all participants have the right to the benefits of the study. And so if there is a positive effect on the experimental group, but not the placebo group, you need to make sure that that placebo group also has the opportunity to receive that treatment as well to fulfill your ethical duties. There's also the expectancy effect. This is experimental bias. This is the impact of the experimenter's bias on the outcome of a research study. Researchers could potentially treat participants differently in the various conditions of the study. So they might be, let's say, more nice to the experimental group than the control group, or they behave differently, which can affect how participants perform. And when recording behaviors, there could be subtle differences in how the researcher interprets and records them. Again, if we're doing observational measures, that if a researcher wants to make sure that they want to find a significant difference between the control and experimental group, they may inadvertently actually report more differences than are actually there. So there's a couple ways to minimize this. Experimenters need to be well trained and should practice behaving consistently with all participants. So it's just again, that rote behavior over and over to train themselves, and then run all conditions simultaneously so the experimenter's behavior is the same for all participants. So that's where the placebo and the control and the experimental group would get the treatment all at the same time or lack thereof. Thus, they're exposed to the researcher's same behavior. Their automated procedures can also minimize expectancy effects in that the researcher is kind of removed. And this also rolls into single blind and double blind experiments in that with single blind, the participant is unaware of whether a placebo or the actual drug is being administered, which is pretty much every experiment. And then there's the double blind in that neither the participant nor the experimenter know whether the placebo or the actual treatment is given. So the experimenter does not even know which group a participant is in, thus decreasing the possibility of that expectancy bias. So when it comes to final planning considerations for an experiment, research proposals are usually written after the researcher has put considerable thought into planning the study. Sometimes there's a pilot study. This is a trial run with a small number of participants to determine, again, the reliability of a measure or how efficacious the study actually is. There's a manipulation check, and this is the attempt to directly measure whether the independent variable manipulation has the intended effect on the participants. So to make sure it's actually creating that effect. And then there's usually a debriefing session that's going to provide an opportunity for researchers to interact with participants after the data has been collected in order to get their feedback and answer any questions or alleviate any concerns that participants might have. When it comes to pre-registration, this is a process by which researchers identify and articulate their research questions, hypotheses, and analysis plan for their studies before they collect and analyze data. A response to the open science movement, which emerged as a result of threats to the integrity of modern science, has now been pursued, and the final planning consideration would be to pre-register a study and analyze plan in an open access archive. So that provides that provides accessibility to other scholars, academics, and the public. There is one, and I will put a link to it because I am not recalling it exactly, but what it has entire outlines for studies as well as IRB approvals, and it provides all basically the backbone of an entire study for, again, it's open access. You would be able to read it, I would be able to read it and review what the process actually was. Then, once data are collected, statistical analyses are going to be carried out to allow the researcher to examine and interpret the pattern of results to determine whether there's a significant difference between groups. And this will help the researcher decide whether there really is a relationship between the independent and dependent variable and how strong that relationship is. Now, when it comes to communicating the results, there are meetings, there are conferences, regionally, state, regional, national, and internationally to present findings to other researchers. This could include like the meeting of the APA that occurs every year. And also research papers submitted to journals are subject to peer review. So if you were to submit your research to a journal, it would need to be peer reviewed by other academics and researchers to ensure that it is a valid and thorough study. And as many as 90% of papers submitted to the more prestigious journals are going to be rejected, which is it's not great, but it's also it's not bad, because what that means is that they're only taking the very best research, most impactful research, the most thorough and ethically conducted research, and that's what's getting published in these journals. So, with that being said, I hope that gives you an idea of how experiments are run. Again, if you have any questions or concerns, feel free to let me know, and I hope you have a great day.